Early Drosophila embryos undergo 13 rounds of rapid nuclear division before enclosing each nucleus into an individual membrane-bound cell. In the initial slow phase of this cellularization process, the plasma membrane invaginates and forms a series of cleavage furrows, whose leading edges are interconnected by a hexagonal actomyosin network. Once the cleavage furrows have grown past the base of the nuclei, the second, fast phase of cellularization sees the actomyosin network transform into individual contractile rings, which seal off the cells at the base to form an epithelial layer called the blastoderm. Stefano De Renzis from the EMBL in Heidelberg, Germany, is interested in how phosphoinositide lipids might coordinate the processes of membrane growth and actomyosin contraction during blastoderm cellularization.
We knew that phosphoinositides are important for controlling actin dynamic at the plasma membrane. However, we didn't know what role they play in vivo in the context of a morphogenetic process like cellularization and tissue morphogenesis.
De Renzis and colleagues, led by postdoc Alessandro Reversi, began by injecting Drosophila embryos with membrane-permeant phosphoinositide analogues. Once inside the embryo, these lipid derivatives, developed by Carsten Schulze's laboratory at the EMBL, are unmasked and inserted into the plasma membrane, rapidly elevating the levels of specific phosphoinositides.
We started with several compounds, but the two that gave the more striking effects were the PIP2 and the PIP3. And what was interesting is that these two phosphoinositides were having complementary impact on cell shape and actomyosin contractility.
Injecting early embryos with membrane-permeant PIP2 caused premature contraction of the actomyosin network during the slow phase of cellularization, generating shorter cells whose nuclei get trapped in the actomyosin rings and pushed to the embryo's interior. Injecting membrane-permeant PIP3, on the other hand, caused the actomyosin network to disassemble during cellularization, resulting in the formation of abnormally shaped multinucleate cells. Reversi et al. Therefore examined the dynamics of myosin II, which concentrates at the leading edge of the cleavage furrows as cellularization proceeds.
The level of myosin increased upon injection of PIP2 as much as they do under normal cellularization conditions from the slow to the fast phase of this morphogenetic process. And the myosin become more stably associated at the furrow when we increase the concentration of PIP2. So it's not only more myosin, but the turnover of myosin is different, meaning that more myosin molecules are stably associated at the plasma membrane.
This would explain why PIP2 promotes actomyosin contractility. To investigate whether the phosphoinositide is required for actomyosin constriction, Reversi et al. Used a protein dimerization system, in which the small molecule rapamycin is injected into fly embryos at different time points to induce the recruitment of a lipid phosphatase and the rapid turnover of plasma membrane PIP2.
Injection of rapamycin prior to the beginning of cellularization causes a complete disassembly of the actomyosin network and disassembly of the furrow. However, if we allow cellularization to start and then injected rapamycin and therefore reducing the level of PIP2, cellularization could proceed, but late during this process the actomyosin network did not contract and therefore cells remain open at their base. So this experiment really allowed us to demonstrate that PIP2 is required at two different stages for actomyosin contractility.
But what inhibits actomyosin contractility during the initial slow phase of cellularization? Reversi et al. Found that PIP3 is only present at the leading edge of the cleavage furrows during this early phase, and that blocking PIP3 production with the PI3 kinase inhibitor Wart-Manin induced premature contraction, similar to the injection of PIP2. The same phenotype has also been observed in embryos lacking a protein called bottleneck.
And what we found is not only bottleneck and PIP2 have a similar morphological phenotype, but they are associated exactly to the same type of alteration in myosin dynamics and turnover, meaning that in a bottleneck mutant we observe higher level and increased stability of myosin at the forward. And what we found is that bottleneck interact very specifically with PIP3, and that PIP3 is required to control bottleneck localization at the leading edge of the furrow where the actomyosin network is assembled. And so this experiment together suggests that PIP3 acts upstream of bottleneck, and PIP3 and bottleneck together counteract the contractile promoting activity of PIP2.
To investigate how bottleneck might inhibit actomyosin contractility, Reversi et al. Overexpressed the protein in HeLa cells.
And what we found actually was very striking. Bottleneck localized to filaments in HeLa cells, and this filament resembles stress fiber, and the level of actin on the stress fiber was drastically upregulated. And the third important result of this experiment was that the stress fiber became completely resistant to actin depolymerizing drugs. So this data suggests that bottleneck act as an actin stabilizer, presumably as an actin cross-linker, that limits the turnover of actin filaments.
In contrast, bottleneck didn't upregulate the amount of myosin on these stress fibers, thus producing filaments with a high actin to myosin ratio and presumably low contractility.
So our model suggests that the level of phosphoinositides, particularly phosphoinositide BIP2, are stable during cellularization and are required for the assembly initially of an actomyosin network, and that this network needs to be kept on hold during the phase of plasma membrane expansion. And this is controlled by the presence of another phosphoinositide of PIP3 and recruitment of the bottleneck protein. During fast phase, the level of PIP3 is decreasing, bottleneck is degraded, and now the contractile promoting activity of PIP2 can be unmasked, and therefore the network contracts, closing off the cell at their base, ensuring the generation of cell with the proper shape and length.
Increasing PIP2 levels or suppressing PIP3 or bottleneck causes premature contraction and abnormally shaped blastoderm cells.
We think that this could really be a general mechanism controlling the timing of actomyosin contraction during morphogenesis, and we are now actually testing this hypothesis by addressing the role of the ratio between the plasma membrane phosphoinositides in other morphogenetic processes, such as ventral furrow formation, for example, where also the timing and contractility of the actomyosin network need to be coordinated with the changes in plasma membrane area.
De Renzis and colleagues also want to further investigate how bottleneck inhibits actomyosin contractility, but in the meantime, you can learn more about how the ratio of plasma membrane phosphoinositides regulates cell shape during Drosophila morphogenesis in the paper by Reversi et al., published in the May 12, 2014 issue of the Journal of Cell Biology.